First, I'll illustrate bonding and specificity in DNA. I made the phosphate backbone and started with deoxyribose. Each had one pipe cleaner, 5 beads, and 2 gold ringlets. I put 5 beads on each deoxyribose to represent our carbons and a gold ringlet by the 3' and 5' carbons to help me remember where the phosphates are bonded. 1' is where we will attach the base, 2' is where we can tell if it is DNA or RNA by whether or not an oxygen is present. In our case, there is no oxygen because we're making DNA. 4' carbon is where the 5' carbon branches off. Then I added nitrogenous bases to the 1' carbon of deoxyribose. I'm using old Easter eggs as the bases, with pink representing complementary bases adenine and thymine. And these are bonded by 2 hydrogen bonds. And blue represents complementary bases cytosine and guanine, which are bonded by 3 hydrogen bonds. Since pyrimidines have 1 ring and purines have 2, we'll have the longer sides of the eggs representing purines and the shorter sides of the eggs representing pyrimidines. Now, if we look at a double-stranded DNA, each phosphate molecule is bonded to the 3' and 5' carbons, so the connections of the sugar-phosphate backbone are called phosphodiester bonds. Notice how one DNA strand has oxygen pointing up in the deoxyribose, and the other has oxygens pointing down. This resembles an anti-parallel structure. I chose Easter eggs in order to demonstrate that the purpose of hydrogen bonds are for specificity, so all base pairs are complemented. However, this model doesn't illustrate how the structure of DNA is supported. Hydrogen bonds between the clothespins are not the force that holds 2 strands of DNA together. Instead, it's due to the hydrophobic interactions of the uncharged bases. Since they are uncharged and hydrophobic, the backbone is negatively charged and hydrophilic, the flat bases can lay horizontally and stack themselves in a space-efficient and low-energy manner. This is called base stacking, which creates more hydrophobic interactions in this inner area, making it more stable. The negative charge of the backbone is destabilizing and hydrophilic, but the salt neutralizes the negative charge, making it more hydrophobic like the bases. This increases the melting temperature because more energy is needed to separate DNA that is made more stable with salt. To illustrate this, I'll push a pipe cleaner that is hydrophilic and saturated with water through the middle of an oil blot, and the difference of polarity will break the oil blot. But if I take a pipe cleaner that's saturated in oil and decrease differences of polarity, it's slightly more difficult to do. Major groove is a larger gap between strands, while the minor groove is smaller. DNA is right-handed if it looks like a spiral up a staircase on the right-handed side, and it looks like it's left-handed if the handrail is on the left side walking upstairs.